Okay, so we're back, and we're talking about acyclovir, and this drug mimics deoxyguanosine, which is used in DNA synthesis. So what that does is the virus ends up using the drug instead of deoxyguanosine to build DNA, and that renders the multiplying of the virus, futile. Um, it's not going to work if it doesn't have the right DNA made. So that's administered topically. Unfortunately, using this technique does not usually obliterate the virus completely, therefore it returns, resulting in flare-ups. This is used in genital herpes a lot. Occasionally, there are some severe cases that have acyclovir resistance, and so ganciclovir is used. And I'm going to show you the two structures of those drugs right now. So here's acyclovir, and this is kind of like the nitrogenous base region, so this mimics the guanine piece. And then look at ganciclovir, and you'll see that there's a very small difference between the two molecules. I'll go back and forth a couple times. Essentially, most of the structure is conserved. The difference is in a seemingly unimportant piece over here next on this carbon, right? But the difference is, so it can still have the same impact because the guanine piece is kind of over here. But over here you can sort of avoid resistance and recognition by enzymes in the body by changing the structure just slightly, and you can change the whole lock and key thing. So once again, we're seeing that same story in action. So that's why ganciclovir might be used. So that's one method. Alter the cell's genetic material so that the virus can't use it. One other way to do it is to prevent the new viruses from leaving the cell, and that is amantadine is used for the treatment of flu, and this is accomplished by inhibiting an enzyme that the virus uses for sticking to the host cell wall as it leaves, and so the new viruses can't leave the cell as well. And this actually is a similar technique that is used for some of these newer antivirals like oseltamivir, which is Tamiflu, and zanamivir, which is Relenza. Those are the two key drugs used for flu treatment. So take a look. Here is amantadine. Amantadine is a very simple looking molecule, although it's got 3 fused cyclohexane rings, so it's a little bit 3D. But essentially, it's this little hydrocarbon piece with an amine on the end. That's it. It's a pretty simple molecule. But other similar class of drug molecules, like this is Relenza, and Tamiflu is right here. There's Tamiflu and Relenza. Not exactly the same, but there is some definite similarity in structure here. Okay, and those work by inhibiting the neuraminidase enzyme. So take a look. That's a pretty good explanation of it in the Relenza mechanism of action here. If you look down here, you'll see that it works by binding to the active site of the neuraminidase protein, rendering that virus unable to escape its host cell and infect others. So you can imagine that if you can't increase your viral load through replication and release, you can reduce the amount of time that people experience symptoms. And so they actually have seen zanamivir found to reduce the time to symptom resolution, in other words, the time it takes for you to feel better, by about 1.5 days, as long as your therapy is started early. And you may have heard that before, that you have to start the process early with these influenza drugs. And the reason for that is probably makes sense to you at this point. If you wait until too late in the process, then your viral load is already very, very high, and that's not going to be helped by these drugs that stop the virus cells from getting out of their hosts. You've already got the viral load that is causing your awful, awful symptoms. So the earlier you start, the better off you'll be, because then your viral load never gets that high, and so it resolves faster. So these makes total logical sense as to why these drugs work the way that they do. So do write some of that stuff down. If you need to go back and replay the last 45 seconds, you certainly can go ahead and do that. You don't need to draw the Tamiflu and Relenza structures. Oseltamivir is in your data booklet. In addition, we're going to come back and talk about how Tamiflu is synthesized, because that's going to have some other implications for other things we need to talk about. So the last piece of the antivirals is about HIV and AIDS. HIV is the virus. AIDS is the condition that results from the progression of the virus. Does anybody know what kind of virus causes AIDS? What kind of virus is HIV? Press pause for a second to talk to your groups for 15 seconds, and if you don't know it, you're about to find out anyway, so see if anybody knows. Okay, we're back. So hopefully you paused it, but whatever. The name for that virus is a retrovirus. And retrovirus does not mean it's a virus with bell bottoms on. It means that it only has RNA, no DNA. Now, it is really hard to treat HIV because it is a fast mutator. You may have heard very fast mutation rate. So while you might try and target a certain virus, the actual virus that is active at that moment might be slightly different, and all of a sudden it will not be treated by the drug. So how does the virus work? The virus works by invading certain types of leukocytes. Leukocytes, by the way, are white blood cells. Let me turn on insert here. Okay, those white blood cells are called T helper cells. There are lots of different kinds of white blood cells, but the ones that we care most about are T helper cells. It's capital T. Specifically, they focus on what's called CD4 positive T helper cells. Those are T helper cells that express the CD4 receptor. CD stands for cellular differentiation protein, but the CD4 receptor T helper cells are the ones that are impacted by HIV. These receptors are responsible for activation of the immune response, which of course, as you understand, I'm sure from previous experience in different classes, that is the effect of being infected with HIV, is that your immune system, and you should write that down here, your immune system is affected and it can really essentially go to crap. So how does it actually work? Well, when a cell is invaded with HIV, the virus produces viral DNA from the RNA that's a bad typo here, in the virus, using an enzyme called reverse transcriptase. And just like all enzymes, the ACE tells you what it does. It does reverse transcription. Normally you think of DNA being transcribed to RNA in the first process of cellular replication. Here, it's RNA being copied up to make viral DNA. So it happens kind of what we would say is in situ. It happens in the moment rather than separately. There's no DNA to start. Not surprisingly, this is, of course, the reverse process of what happens in normal cells. But that's actually really good in a way, because if this is a virus-specific process, then maybe a drug can target just that. So the treatment for HIV is predominantly a drug called AZT, which inhibits reverse transcriptase. So instead of targeting the virus itself, it's targeting an enzyme within the virus. Now, since that enzyme is only used by retroviruses, even regular viruses don't have to use it because they have DNA. Normal cells end up being unaffected. There's a real advantage. Now, due to the speed at which this virus mutates, unfortunately, and a close link of metabolism between the cell and the virus, you can't go and attack the metabolism aspect because there's too much similarity. Cures, okay, although, look, let's be honest, HIV, while you can't really consider it a chronic condition now, the survival rates and the survival times, I should say, are continuing to get longer, which is a wonderful thing. But vaccine development is tricky because if you have a mutating virus, it's very hard to figure out which proteins you want to target and get your immune system to recognize. So cure and vaccine development remain very large challenges, although, as you might imagine, they are still very active areas of research. So there's your antiviral video, I guess antiviral videos. But thanks for watching, and now I think you're ready to move on. All right, take care.